related to the vaccine development. Okay, thank you. So I will going to talk about the vaccine development and especially how different options are there for a vaccine to develop a vaccine. And particularly I will be using the coronavirus case, but because in that time, this is the most recent thing. And this article which I am sharing right now on your screen, I will put this thing on your Moodle as well. That is an article which actually covers the starting story, the race which actually started during the mid of 2020. That was the race to get corona vaccine. Who will going to get that thing first? Initially, I thought that I should give this thing as an assignment to you guys, but this is too sensitive and too important stuff. So, and although I believe on you guys, but still I want to speak on these things by myself, okay? Because I want you guys to understand that when something like this coronavirus, a pandemic level problem is there, then what usually pharmaceutical companies was trying to do at that time, how they think about the pandemic at that time, and what are the options they actually tried at that time, and which one of them. You should be able to know that their names, so you should be able to track down which one of them becomes successful now, because now we know which one of them is a success story and which one of them is a second tier type of success and the third tier type of success. At that time, nobody knows. Whatever I'm sharing with you guys is like somehow in 2020. So nothing was confirmed at that. We never know at that time that Pfizer will going to lead in terms of quality of a vaccine at that time. So let's start that what happens. So before the normal things, I will going to tell you about the concept of the vaccines. Traditionally, I'm sure you should be able to see my cursor as well, okay? The students on the online, please stay with me, okay? This is going to, this is one of the way to build your concept related to the vaccine development. And I think the people who have studied molecular immunology, it might be comparatively easier for them because I have talked a lot about these things with them in their class as well. But just give margin to everyone, okay, in our classroom. So just be more patient and please have more questions, okay, and push me to speak more on the things which you fail to understand. Traditionally, the vaccine is something which gives immunity to a human, which gives immunity to an organism against a certain pathogen. Traditionally, at this time, we are only specifically talking about the coronavirus. So we will study all the story by putting the coronavirus in this in the perspective. The coronavirus is a family of different types of viruses. Since their structure looks like that it is made up of some sort of crown over there, the spike proteins, which are very famous, they are present on their outer layer. They look like a crown, so they are called as coronavirus. A lot of families of coronaviruses are out there. Most of them are flu viruses. Some of them actually manage to jump between organisms earlier. So that is why it's a kind of little concern for the humans, because it is called zoonotropic shift, shifting from one organism to another. I will explain how actually it happens when a virus actually shifts from one organism to another. You might remember the bird flu virus, H5N1 virus. You might remember the very famous MERS virus. Those were the viruses which actually transferred from the other organism to the human. So what that means in an easier term is that the virus is capable of infecting a certain host cell once one of the viral protein is able to dock itself with the host surface protein. Full stop, period. Is that clear? A virus is able to infect a host organism only when a virus protein is able to dock itself with one of the protein present on the host cell. So in that case, coronavirus, the spike protein is the one which is supposed to dock itself on the host cells. Now, what if the host cells are the liver cells? In that case, the coronavirus will have to travel through the respiratory system, then have to diffuse into the blood system, then they have to face the immune cells, the first line of defense. They have to circulate into our body until unless they came across the liver. And that happens in case of HIV and HCV and HPV. Not HIV, sorry, HPV and HCV. Hepatitis B and C viruses. Because their surface proteins are capable of binding with the receptor present on the liver cells. So it is like harmless for all the other organs of the body. Even if someone like dropped the HCV virus into kidney, into the brain, or into any other cell, it is like harmless because it will never be able to attach or attack or infect the host cells. So that is why it is very critical and very sensitive and very important for any virus to find the host cell. The coronaviruses are supposed to be very infectious. Remember, having been more infectious means a virus is able to propagate itself faster. The virus is able to transmit itself faster to the other organisms, to the other members of the same species. The internal structure of the brains, like the trachea, the bronchioles, the alveoli, and the air sacs, everything is carrying the air. And that air has no separation. That air is exactly 100% the same air which is present outside. So nature has created our respiratory system in such a way that there should be no obstruction, zero obstruction until the very end. आपको पता है, you know, if there is a little obstruction somewhere inside the trachea, somewhere inside the bronchiole, it started causing hell of the trouble, coughing and all these trouble, blah blah. So nature has created our air passageway completely unobstructed. So whatever is in the air, it's in the lungs. If the air contains like certain contaminants, believe me, it is present in the lungs as well at the same time. So imagine a coronavirus is there in the lungs. Oh, sorry, in the air. That means it is in the lungs as well. If it is present somewhere there, it is present there as well. And unfortunately, the target host cells of the coronavirus is the cells which are creating the air passageway. And how it is possible they can infect the cells? If they are able to dock themselves with a certain protein. The spike protein here is able to bind with the ACE2 receptor of the cells. And this ACE2 receptor is present in the cells which are constituting the air passageway. So that means it's like just an open space, a welcoming, open gateways for the viruses. If you ask me, how easier for the virus to infect the humans? I mean, it is a peanut for that virus to infect the human because the virus will not have to travel. One thing, the virus will not have to face our immune system. Our immune system kahan hai? Within the cells, within the blood, and the virus will not require to meet with the immune cells. So that means it is outside the human body. So that is why it's like undefended cells. So this is the reason the coronavirus is more infectious as compared to the other diseases. Now you can ask one more question. The other story is simpler. The point number 2, 3, 4 are quite simpler. You can ask me another point, sir. This ACE2 receptor, is this present in the other cell type as well? The answer is yes. It is present in the other cell types. So does that mean the coronavirus will going to infect to other type of cells? The answer is yes. Initially, in the first year, a lot of studies have been done when people have reported that it can affect the lungs, it can affect the kidneys. It was reported at that time, and that is because of this hypothesis of the presence of ACE2 receptor in the cells. So all those cells which are carrying the ACE2 receptors are like the direct targets of the coronavirus. But other cells are present beneath the layers, away from the surface, and there are a lot of defense things are present between them. So the most vulnerable stuff out of all these cells are the cells of the air passageway. It is like outside, it is present like outside, like completely exposed cells. So once this ACE2 binding is there, virus कैसे infect करते हैं? बच्चों का खेल है. I am not talking about these things. I want you guys to brainstorm on certain important things which might be the research topics in the upcoming years. हैं? Let's see. For example, पाकिस्तान के अंदर कोई तबाही नहीं हुई, इंडिया में लोग तबाही के शिकार हो बहुत ज्यादा. USA, most number of cases, number of deaths, number one. Then India, then Brazil, something like that. UK and France. These are the people like top listed countries. European and Americans. They actually bear the brunt of the coronavirus. And then after that, later on, India was the target of that. If somebody is working and somebody is thinking as a thinker and try to find out the difference that why certain countries like Pakistan and other countries, they actually not affected that much, is they are more responsible. Anyone agrees? We are more responsible than Indians? I don't think so. It's like this culture is same, the lifestyle is same, the population density is same, the nature of interactions are same. So we are like more or less like a similar sort of people. So why, if something is like created a havoc out there, it does not do anything to us. Hamari 60-70% hospital Allah ka shukr hai, okay? I'm not, I don't want to kaisa hoye, Allah ka shukr hai kaisa nahi hua, but as a scientist, as a researcher, like as a thinker, we need to find out the reason. How it is possible that the people from the same area or from the same planet, some of them are more vulnerable as compared to the other one. So my search or my logical thinking should be landed on the point where virus and cells interact with each other, and that is what we call as the ACE2 receptor. So I might be interested, how about analyzing the ACE2 receptor of Pakistani people and Indian people and USA people? How somebody should create a project out of it to find the reason why this virus failed to produce mass killing in Pakistan, while it was like creating a trouble in the other country. So something is different. It might be genetically different. If you think that it is because that we are more immune, the answer is no. We are not more immune. Somebody told on the other day to me, sir, we are living in very bad conditions. We are inhaling pollutant air. We are like eating pollutant water, food, drinking pollutant water, a lot of contamination and toxins. So our immune system should be very much experienced. The answer is no. This is a complete rubbish concept of immunology, and many people have. मैं बहुत शाने लोगों को देखा उनका ये concept. Okay, put yourself in trouble, and you will be all fine in the future. The answer is no. This thing does not work. इसका आसान जवाब ये है. Somebody asked me. इसका आसान जवाब ये है. If you want to see that our immune system is better or not, just see our life expectancy. You see how long we live. कितना जीते हम? कितना जीते हैं? राबिया मैंने अपना नाम ठीक लिया. कितना जीते हम? हम एक बार जीते हैं, एक बार मरते हैं. कितना जीते हम? 60-70s. एवरेज एज पाकिस्तान की कितनी है? I think somewhere between 60 to 65. I'm not sure exactly, but you can guide. Average age of European countries is more than 90. Average age of Japan is almost 100. Korea is 99, almost 99, 100. China is still above 90. So where we are living, 30-35 years less than those people. कौन ज़्यादा immune है भाई? कौन सी दुनिया में रहते हैं हमें? So we, as a thinker, as a researcher, now we need to be very logical in our answers, in our thinking process. So coming back to the point that these are the things where you need to put your energy to know, okay, why the difference exists in different world population in terms of coronavirus. Okay, so what happens after that is quite simple. You know that the virus enters into the body, it releases this RNA, and coronaviruses are RNA-based viruses. This RNA is then captured by the translation machine and started translating the things. And then reproducing the viruses. It's quite simple. Now, next thing is that how different types of viruses have been created. How different ways are out there. Generally, initially, I'm just sticking with the virus, okay? No bacteria, no microorganism, no fungus, nothing else. Just viruses or coronavirus only. When that happened at that time, the sudden expansion of the disease in different parts of the world, people started working on two fronts. The one is what we call as the weakened viruses. The other is what we call as inactivated viruses. Weakened viruses to make viruses a little weaker and then use it as a vaccine. Remember, if the viral structure is retained, the more viral structure is there in a vaccine, the more potent that virus will be. देखो अगर वो वायरस की तरह की होगी बिल्कुल और जितना मैक्सिमम वायरस का आप लुक बचा के रखेंगे उसका, your immune system will going to react, will going to consider that thing as a vaccine. So that means this is a seesaw sort of calculation you have to do between two factors. One, it should be more similar to the virus. And number two, it should not harm the person's body as well. So you need to remove certain things which can make the virus impotent, but it will allow that virus to stay as a virus. That is what we call as the weakened version of virus. For example, if the viral genome contains certain genes, some of the genes help that virus to produce the protein for the penetration. Some of the genes help the virus to produce some earlier proteins. Virus में सारी proteins नहीं होती, वो इतना चालाक है, it just produce some initial proteins. और वो अपना RNA polymerase reverse transcriptase साथ लेके आता है. The virus, the RNA viruses, they carry their own reverse transcriptase. That is why we never able to find there is a reverse transcriptase earlier. जब तक हमने virus को खोज नहीं लिया. It was not there in humans. It was there in the virus. दुनिया जहान, the world was thinking about the central dogma: DNA, RNA, proteins. The virus was working entirely oppositely. RNA, then DNA, and then start doing the job. So RNA, keeping an RNA is an easier choice, but keeping an RNA is useless because there is not a single enzyme in any person's body, in any species, which can utilize RNA molecule. So that one important critical thing is reverse transcriptase, which can convert that RNA into the DNA. So the virus brings its own reverse transcriptase as well. The rest of the things are fine. You can get the amino acids for translation. You can get the nucleotides from the cytoplasm. You can go for the synthesis of DNA without any problem. All the material is there. But what was not there? Some initial proteins and the reverse transcriptase. The minimum agenda. So now think about that viral genome. It contains all the critical things. Some of the things are required for the reproduction of virus, to produce new viruses. Some final things are required to assemble, to create the virus assembly at the end of the day. You know, new viruses are born. So some of the genes are utilized to produce things which can assemble the viruses at the end, virus assembly. But if you clip and chop off those genes from the virus, what will happen? The virus will be able to penetrate? The answer is yes. The virus will be able to, kick start the immune system? The answer is yes. The complete potent working virus is entering into the cells. Whether that virus will infect the cells? Answer is somehow yes. But the definition of infection is not this. The definition of infection is this: from one cell to another. एक cell पे कब्जा करके मार के और आगे जाओ, that is infection. If a virus is able to penetrate into a cell and then dies over there, this is not infection. But that virus will be able to kickstart the immune system, but that will never be able to reproduce. Weakened version of inactivated version of virus is that the viruses are treated with certain chemicals, so that most of the life activities of the viruses, the potential life activities of the virus is deleted. But whatever left was the structure of the proteins. different three-dimensional structure of the protein. And they make sure that the viral structure should stay intact. So all the virus is there. It is like if you like freeze, kill, freeze. The virus will be like that. No life, but frozen with all the structures intact. अभी जितनी वैक्सीन हमारे पास है ना, आपने सबको चेक करना कौन सी कहां से आई है जी. नहीं, ये जो मैं बता रहा हूँ ये नहीं है. जो आप बता रहे हैं वो मेरे दिमाग में नहीं आ रहा, शायद वो हो गया, मैंने पढ़ा. हम जो मैनिपुलेशन करते हैं ना, जो इंजीनियरिंग करते हैं, हमारे पास एक ही चॉइस है इंजीनियरिंग करने की. एंड व्हाट वाज दैट? वायरल जीनोम. This is the life, no? The source of life is the viral genome. Whatever will going to happen after that will be harvested, will be read, or will be extracted out of this piece of nucleic acid. उसी nucleic acid पे लिखा हुआ है कि when a time comes, there will be a time when the virus assembly will be there, and these genes will going to play a role at that time. And you just clip those things. And a lot of other ways to make the virus impotent as well. What we need is that we need a structure which can mimic the virus perfectly, to cheat our immune system, to assume that a real virus is there. That's one thing. If a complete virus is there, because of the size, structure, the immune system will be triggered. So this is one of the way that it can trigger the immune system. And of course, once the virus is there, it is captured by our body's rangers, which are called as antigen presenting cells. They are like rangers. They actually move from one place to another place within our body. They are different in different organs. So whenever they detect something weird called non-self, something which does not belong to them, whatever which belongs to our body is called as the self, and whatever else, this is called as non-self. The viral proteins are not present naturally in our body, so those things are called as non-self. The antigen presenting cells will going to capture them and then share that information with the next member. These are called as the T cells. Actually, it is written the T helper cells, but it should be like naive T cells or the T cells. That is fine, but it is fine. So nature paper है, इसलिए वो अगले steps का बता रहे हैं. They are not starting it from the very basic. They actually sharing the step which activate the beta cells. इन्होंने ये structure पूरा करना था. I'm sure that is why they use the term T helper cells here. Traditionally, a cell which is required to be communicated here, it should be a naive T cells, a T cell which does not or never came across any problem or any pathogen. So this information will be passed on to the T cells. T cells will proliferate. It will proliferate and produce effector cells. One of the effector cell is called as the helper T cell. Just throw a skip per game, but that's the same thing. Now this helper T cells, which is activated as a result of the virus, that helper T cell will going to activate the beta cells, and beta cells will transform themselves into the plasma cells, and of course they will start producing the antibodies. These antibodies are produced under the instruction of the beta cells, and that beta cell is pushed by the T cells, and these T cells are activated according to the information given by the antigen presenting cells. So if I complete that story, that means these antibodies are the perfect answer or the perfect target to the virus particles. They can capture, they can bind themselves with the virus, and they can kill the virus. That's the one thing. Another type of T cell, actually they do not come out of the T cell. The drawing is a little bit confusing, so I'm just explaining it. When the T cells got activated, they produce different types of effector cells. One of the effector cell is T helper cell, and we know the story. The other type of effector cell is called as cytotoxic T cells. More commonly, they are called as CD8 cells. They are the cell killers. They are able to kill the cells. Now, it is a very specific, dangerous, and special job assigned to somebody within the cell. The ability to kill human cell, किसी के पास नहीं है. Life is like the most prestigious thing. You cannot convince life to die. So now you are giving someone a license to kill. Giving us, ये cytotoxic T cells होते हैं, they are able to kill the cells. Kill the cells, not the virus. Please understand this point. They are designed to kill the cells. What type of cells? Infected cells. Because they are raised out of the information of the bacteria, of the virus. So they are able to kill all the cells which are carrying the viruses. So two things are doing. The one is killing the infected cells. So the virus should not be able to receive or find the host cells. And the other is like a mass removal of the viruses with the help of antibodies. Humoral immunity. Producing antibodies within the blood. ताकि virus कहाँ पे जहाँ पे भी ऐसा. ये बहुत बड़ा function जो है execute हो रहा virus के बाद. That is a basic how they can actually infect the cells. Now think that what happens in case of different types of vaccinations. But let me tell you something more before that thing. Look at this data. And just अगर आप लोग मेरे साथ ही है ना, साथ रहना ओके. मैंने ओपनिंग में इस डेटा के बारे में बताया था कि that was the time when nobody knows what will going to happen. And this is an excellent time to review their strategy. इनमें से कौन-कौन समझदार था, कौन-कौन समझदार नहीं था. वो लोग कहते हैं ना कि उन्हें अकलमंदी ਦੇ ਨਾਲ ਉੱਥੇ ਪਲਾਟ ਲੈ ਲਿਆ ਸੀ ਅੱਜ ਉਹਦੇ बड़े पर. हम लोग अक्सर अपने अम्मा से खिला करते हैं ना. क्योंकि अब्बा जी अक्सर एक स्टोरी सुनाते होते हैं कि उस समय प्लाट 15,000 रुपए ਦਾ ਸੀ मैं नहीं लेता. बंदा कहता ਤੁਸੀਂ ले लेंगे आज ਅਸੀਂ करोड़पति हो जाना. So at that time, look at the comparison between different type of viruses. So the strategies were there that we will going to create, the pharmaceutical companies were started creating the vaccines with the help of inactivated or weakened viruses. So two are there, this which I have told you a little while ago. Please remember the colors coding as well. And then the viral vectors. The viral vectors which are used in vaccine development are of two types. Number one, actually जो हमारा content था natural medical biotechnology का वो बहुत I didn't like it, so I changed it. Sorry, updated it actually for you guys. So the one is the replicating one, the other is the non-replicating one. अच्छा आपने नहीं सुना होगा कि क्या कोई ऐसा वायरस का वेक्टर हम इस्तेमाल करते हैं जो जाके रेप्लिकेट भी करता है. You might remember using viral vectors. जीन थेरेपी में पढ़ा हमने. CRISPR-Cas टेक्नोलॉजी में उससे पहले कि कैसे सेंड करेंगे वायरल वेक्टर यूज करेंगे. The same thing. But those viral vectors are supposed to be delivered within the cells. They become integrated into the host genome. Some of them are that which stays in the cytoplasm, they replicate, and they help in the translation and production of new things. Some of them do have the capacity to replicate, the other, they don't have the capacity to replicate. Remember, all these factors are like plus and negative points of the vaccines. Agar ek vaccine is both potent ho, the vaccine is very potent, but it is made up of replicating viruses. Market करना मुश्किल हो जाएगा ना, लोगों को बेचना आसान हो जाएगा. At the same time, if another company raise a vaccine which is somehow similar in potency, but that was based on non-replicating virus, but non-replicating virus vectors, तो उस कंपनी के लिए इसको मार्केट करना आसान हो जाएगा, वो जीत जाएगी नाम से. If we are more safer, we are more better, we are using the better and safer options as compared to the other company. So all these companies are like taking chances in different areas. And you know the most more difficult or I must say the easier chances were that the people choose the easier route as well, because they just want to create something, because they know जो पहले बनाएगा वो कमाएगा. It was like that. जो पहले बना लेगा वो पैसे कमाएगा. But there were people who took who played a gamble at that time, according to me. Look at that. Those people who are working on DNA and RNA, using DNA and RNA or using nucleic acid as a vaccine. Pretty new concept. Using DNA was supposed to be the best candidate, but they have failed. DNA stable होता है, deliver करना आसान है, रखना आसान है, transport करना आसान है. So how about creating a DNA vaccine? Exciting concept था ना, but it didn't work. It is equally, look at that, the nucleic acid based and then the RNA based. that many number of people or number of companies are involved in trying to create an RNA-based vaccine. It's not only the Pfizer. There are other companies who were trying at that time. Pfizer भी करता ना फेल हो जाता अकेला. मैं बताता हूँ कैसे फेल नहीं हुआ. And then there are protein subunit based viruses. They are virus-like particles. Somebody tried to create an artificial virus to mimic the coronavirus, and they have experimented as well to check whether the cells respond to the artificial particle in the same way or not. And they actually put and embed, some of them put and embed the spike protein-like things. Well, they created a virus particle, viron, put the spike protein-like things. So the body should consider this thing as a virus and start reacting and start producing. People tried wonderful things at that time. DNA and RNA based. Now we know which one of them was the most famous. आज इन सब में से जबरदस्त कौन रहा है भला? जिसने क्या सिलेक्ट किया था? जिसने सिलेक्ट किया था RNA based virus. Pfizer at that time. आप मुझे बताएं कि साइनोवैक आपने लगवाई है? मैंने लगवाई है बस. किसने लगवाई है? मैंने लगवाई है. मैंने लगवाई है. मुझे बताओ इनमें से कौन सी वाली है जो मेरी बॉडी में इस टाइम काम करने की कोशिश कर रही है? And Sinovac and Sinopharm are almost same. Okay. G. Okay. Anybody from the online?
Protein based, sir.
Proteins based, subunit based. Good. I will tell you a website which will explain you कि कौन सी वैक्सीन कैसे शुरू हुई और कहां तक पहुंच चुकी है और उसका कितना डाटा और ट्रायल का जरूरी है आप लोगों के लिए. आप इससे रिलेटेड 3 या 4 वीडियोस हैं, फाइजर से रिलेटेड और वैक्सीन डेवलपमेंट से रिलेटेड, वैक्सीन को ट्रेस करने के तरीके. आप काइंडली वो जरूर देखिएगा. और उनमें से एक जो है वो 2 उर्दू में है और एक इंग्लिश में है. वो मेरे क्लास का लेक्चर है, तो इसमें पूरा रिव्यू दिया हुआ है FDA के हवाले से. बात सारी बात go and just try to spend some time with these videos. These will open your mind a lot about the vaccine development. Okay, let me take you back to the point where we have started. I was telling you the Pfizer at that time was selected. मैं इसी स्टोरी को यहीं पे कंक्लूड करूंगा, आगे बाद में पढ़ते हैं. तो हमने देखा कि इन सारों में से सबसे कामयाब the most successful companies were the ones which have selected RNA as a vaccine. उनकी वैक्सीन सबसे पोटेंट साबित हुई है. The reason why the Pfizer actually came up as compared to the other companies में ना, look at that, about 20, कोई मामूली बात नहीं है. Pfizer ने 19 कंपनियों को हरा दिया at that time, and they just become the one. They need to create a messenger RNA which can encodes, which can be captured by our translation machinery and produce the spike protein. Full stop. आसान तरीन काम. उनको एक messenger RNA बनाना है which contains the genes and the signals and the signs recognizable by our human system and our translation machinery, to consider that thing as human messenger RNA, capture it and start producing the protein. Whatever the protein, what that protein will be. This real spike protein, accurate spike protein of the coronavirus. Once that protein is produced within the cell, it is like a very weird signal, because that protein is not supposed to be present in human body. It's a very, very weird signal. So once that spike protein is produced right within the cell, our immune system will be triggered. I can tell you it was triggered with a very, very high efficiency, because things are present right within our own cells. So that's why it's quite exponential expression. But the problem is that we are talking about the messenger RNA. Look at the people who are working and doing research in our laboratories, and their friends are trying to extract messenger RNA. And if they are trying to extract messenger RNA from human cells, getting messenger RNA from the bacteria is nothing. It's there. Getting the messenger RNA from human cells is more complicated because it's more protective. The layers are very strong, stable, does not allow simple chemicals to break. And if the chemical allows you to break the cells, that chemical will destroy the DNA as well. and the RNA as well. So all you need to do is to separate the DNA from the RNA within the cells. Notice, if you know these people, सबसे बड़ा problem उनका क्या होता है? The biggest problem these people are facing, that I have extracted the RNA and it is gone. I have extracted the RNA and it is gone. It is gone. They have to do all that experiment on ice, and they need to work very hard to keep the temperature low in order to have the virus at the end of the extraction. I'm talking about extraction, not synthesis. I mean, there are a lot of things and you just need to extract it. It's that much difficult, that much delicate to deal. Now what happens, what we are doing, we want to synthesize RNA and then we want to deliver directly where into the human cells. We thought we will put this thing into the respiratory system. It will be received by the cells. How a cell allow something from the outside to penetrate within the cells? अगर ये इतना ही आसान काम होता है, तो our cells might be penetrating everything from the environment. We are not allowing anything to enter into our cells. You know, cells are too resilient. Does not allow anything to penetrate into the human cells. अगर ये इतनी आसान सी बात होती, how we able to send something across the boundary of the cell membrane? ये कैसे करेंगे? And that is from outside. अगर ये इतना इजी होता तो हमारे सेल्स कोई ना कोई चीजें बाहर से ले ही रहे होते हैं, हम कुछ नहीं बाहर से लेते. Cells does not allow anything. So that stuff is required to be transferred and cross the boundary of the cell membrane, and that is very critical. अच्छा, crossing the cell membrane boundary is very difficult, but thing लेकर किसको जाना है? RNA को. It makes the job almost impossible. RNA is a delicate molecule, and number two, it needs to cross the cell membrane. That is where the BioNTech company came as a player. It is from Germany, and that is a time when these two people their scientists might have a meeting at that time, I guess, like that we are very good in creating RNA, we are very good in creating genes, our molecular biology techniques are wonderful, and you guys are selling something. Biotech company was selling a material which people are using in research to send stuff across the cell membrane. So their specialty was not vaccination. Their specialty was that they are creating some cell membrane like capsules. They are creating some micelles, some cell membrane like capsules, and they were selling it to the other people. People put things in that capsule and then they use that material to send the payload right within the cells. So they were doing that stuff. Vectors को भेजना है आपने ये use कर लें, chemical भेजना है vector के cells के अंदर तो ये use कर लें. So they asked them to collaborate, and then they collaborate. They created the messenger RNA and they created the lipid capsule so that the lipid membrane should be welcoming, somehow welcoming. Still the efficiency is not wonderful, I can tell you. This is a very difficult experiment to send things within the cells. अगर हम controlled laboratory condition में भी करें ना, फिर भी 40-50% से ऊपर efficiency नहीं आती है. ये तो जिंदा बंदे को ऊपर करनी है. It's not a laboratory. My body is different. The other person's body is different. The other person's body is different. Somebody who is sending, somebody who is administering, he is different. She is different. Experiment में तो हम paper में लिखते हैं ना, एक चीज इची बिची, everything should be same. यहाँ पर तो इतने सारे variable हैं, जितने लोग हैं उतने variables हैं. So it is like an experiment which is bound to end in a failure. But they believe that even if we ended up like 4 to 5 to 10 to 15% efficiency, these number of cells are good enough to produce a potent response for the vaccination. And they have made it, okay? I will go to talk more on these things. I wish that this should help your mind to think beyond, okay? Think beyond borders, out of the box.